This is part 2 for the digestive system lecture, looking at the various parts of the digestive system. Today we're starting with the pharynx. So when food is first swallowed, it's going to pass from the mouth into the pharynx, also known as the throat. This is going to be a funnel-shaped tube that's going to extend from the external nares of the esophagus, and it's going to extend posteriorly to the larynx anteriorly. The pharynx is going to be composed of skeletal muscle, and this is lined by a mucous membrane and is going to be divided into 3 parts: the nasopharynx, the oropharynx, and the laryngopharynx. The nasopharynx is going to function only in respiration. However, both the other 2 parts of the pharynx, the oropharynx and the laryngopharynx, are going to have functions in digestion as well as respiration. Once food has been swallowed, it's going to pass from the mouth into the oropharynx and the laryngopharynx. And the muscular contractions that are going to be occurring in these areas are going to help to propel food from the esophagus and into the stomach. The next part of the digestive system is the esophagus. This is going to be a collapsible muscular tube. It's going to lie posteriorly to the trachea, and it's also going to connect the pharynx to the stomach. The esophagus is going to begin at the inferior end of the laryngopharynx, and it's going to pass through the inferior aspect of the neck, and it's going to enter the mediastinum anterior to the vertebral column, so we can see that here. Then it's going to pierce through the diaphragm and then moving through an opening that's called the esophageal hiatus. This is going to end in the superior portion of the stomach. So we've got our esophageal hiatus ending in the stomach. When we take a look at the actual histology of the esophagus, we can see that, as usual, as you would expect, the wall of the esophagus is going to contain 3 separate layers. There's the mucosa of the esophagus, and this is going to consist of non-keratinized stratified squamous epithelium. The next layer is the lamina propria. This is an areolar connective tissue layer. And then the final layer is going to be a muscularis mucosae, and this is going to be a layer of smooth muscle. Near the stomach, the mucosa of the esophagus is going to contain various types of mucus glands. The submucosa is also then going to contain areolar connective tissue as well as blood vessels and mucus glands as well. And then the muscularis of the superior third of the esophagus is going to be made up of skeletal muscle, while the intermediate third, the middle third section of the esophagus is going to be both skeletal and smooth muscle. And then that bottom or inferior third is going to be all smooth muscle. So it's sort of a transition based on what kind of muscle you'd expect to find based on where you are in the esophagus. So at each end of the esophagus, the muscularis is going to become slightly more prominent, and it's going to form 2 separate sphincters. There is the upper esophageal sphincter. This is going to consist of skeletal muscle, and it's going to regulate the movement of food from the pharynx into the esophagus. The lower esophageal sphincter, also called the cardiac sphincter, is going to consist of smooth muscle, and it's going to be found near the heart. And this is going to regulate the movement of food from the esophagus into the actual stomach itself. Looking further at the histology for the esophagus, the superficial layer of the esophagus is known as the adventita. We've mentioned this in previous lectures. This is found in the esophagus rather than the serosa, like what we would see in the stomach and the intestines. The esophagus has this adventita. This is because the areolar connective tissue of this layer is not going to be covered by a mesothelium. And additionally, the connective tissue is going to merge with connective tissue of the surrounding structures of that mediastinum as it passes through. So the adventita is unique and is definitely different than what we would see in the serosa. This adventitia is going to attach the esophagus to the various surrounding structures and kind of helps to anchor it into place. Looking then at the physiology of the esophagus is going to secrete mucus, and it's also going to transport food to the stomach. It does not produce any digestive enzymes, and it's not going to carry on any sort of absorption processes, so no nutrients is being absorbed in the esophagus. During the esophageal stage of swallowing, there's going to be a progressive amount of contractions of that muscularis that's going to be found in the esophagus, and this is going to help to push that bolus onward towards the stomach. So these propulsive contractions are going to be called peristalsis, and this is a term that we'll use throughout a lot of the GI tract. That brings us then to the process of deglutition. This is the movement of food from the mouth into the stomach, and this is achieved by the act of deglutition, aka swallowing. Deglutition is going to be facilitated by the secretion of saliva and mucus, and is going to involve the mouth, the pharynx, and the esophagus all working together in concert. Swallowing is going to occur in three main stages. We have the voluntary stage, the pharyngeal stage, and the esophageal stage. In the voluntary stage, this is when the bolus is going to pass into the oral pharynx. Next is the pharyngeal phase, and this is going to be the involuntary passage of the bolus through the pharynx into the esophagus. And then the esophageal stage is going to be the involuntary passage of that bolus through the esophagus and into the stomach. So during deglutition, the swallowing is going to start when the bolus is forced into the back of the oral cavity and then into the oral pharynx. And this is done or achieved by the movement of the tongue upwards and backwards against the palate. So these actions are going to constitute the voluntary stage that we have of swallowing. So as that bolus passes into the oral pharynx, that is going to be the involuntary stage, i.e. the pharyngeal stage of swallowing. That's when that's going to begin. The bolus is going to stimulate receptors in the oral pharynx, which are going to send impulses to the deglutition center that's located in the medulla oblongata, as well as in the lower pons of the brainstem. The returning impulses are going to cause the soft palate and the uvula to move upwards to close off the nasopharynx to prevent any food or liquids from entering into that nasal cavity. Obviously, we've all been there when this doesn't work like it's supposed to and you get milk coming out of your nose. But generally, it should help to kind of close off that cavity to help prevent things from getting up into that nasal cavity. In addition, the epiglottis is going to close off the opening to the larynx, and this is going to prevent the bolus from entering the rest of the respiratory tract as well. The bolus then moves through the oral pharynx and the laryngeal pharynx, and then once the upper esophageal sphincter relaxes, the bolus is going to move into the esophagus. The esophageal stage of swallowing is going to begin once the bolus enters the esophagus. So during this phase, the peristalsis is going to help to push that bolus onward down into the stomach. So this is going to happen in a couple of different ways, or because of a couple of different reasons, or in a few different steps. So in the section of the esophagus that's just superior to the bolus, there's going to be circular muscle fibers that are going to contract. This is going to result in a constriction of the esophageal wall, and this is going to squeeze that bolus onwards towards the stomach. Then longitudinal fibers that are inferior to the bolus are also going to contract, and this is going to shorten this inferior section of the esophagus, and this is going to help to push its walls outward so that it can receive that bolus. And then the contractions are going to repeat in waves until that food reaches the stomach, or as that food moves to the stomach. So steps 1 and 2 that we've just discussed here are going to repeat until we've moved that food bolus down to the lower esophageal sphincter, at which point the lower esophageal sphincter is going to relax and the bolus is going to move into the stomach. There, mucus secreted by the esophageal glands is also going to help to lubricate that bolus and help reduce any friction when you swallow, as in the whole process of swallowing. So here we have a handy little table discussing the digestive activities of the pharynx and the esophagus for your studying uses later. So moving on, the next part of the digestive system that we're going to discuss is the stomach. This is a J-shaped sort of enlargement that we have in the GI tract. And this is going to begin at the bottom of the esophagus, and it's going to end with the pyloric sphincter, which you can see here. The stomach is going to serve primarily for a mixing and holding area for your food. So this is where digestion is really going to begin for proteins, and it's also going to continue for the digestion of triglycerides and carbohydrates that are going to be converted from a bolus into a liquid called chyme. This is also going to absorb some substances, so some absorption is going to happen in the stomach. The stomach is going to have four main regions. There's the cardia, the fundus, the body, and the pyloric part. They got real creative with that name there. The cardia is going to surround the opening of the esophagus into the stomach, which you can see here. The fundus is going to be the rounded portion of the stomach that is superior to and to the left of the cardia, which you can see that here. And then the inferior to that fundus is going to be the large central portion of the body, or central portion of the stomach called the body. So when the stomach is empty, the mucosa that lines it is going to lie in these really big folds called rugae. These folds, just like every other fold that we've got in the body, are fantastic for increasing the surface area so that more absorption can occur. Next is the pyloric part of the stomach, and this is going to be divided into three additional regions. So the first region is going to be the pyloric antrum, which we can see right here. This is kind of the bottom part. This is going to connect to the body of the stomach, and the second region is going to be the pyloric canal, which is here. You can see in the image. This is going to lead to the third region called the pylorus, which is going to in turn then connect to the duodenum, which is that last bit of stomach, first bit of the small intestine. The pylorus is going to communicate with the duodenum of the small intestine via a smooth muscle sphincter called the pyloric sphincter or pyloric valve, and the concave medial border of the stomach is going to be called the lesser curvature. You can see that right here. And then the convex lateral border of the stomach is called the greater curvature. Fairly aptly named. So the stomach is going to be composed of the same basic layers as the rest of the GI tract, but there are going to be certain modifications that need to be made in order for it to do its job properly. The surface of the mucosa is going to be a layer of simple columnar epithelial cells. These are called the surface mucous cells. And then the mucosa is going to contain a lamina propria, which again is made up of areolar connective tissue. And then there is also the muscularis mucosae layer. This is made up of smooth muscle. The epithelial cells are going to extend downward into the lamina propria, where they're going to form columns of secretory cells called gastric glands. Several gastric glands are going to open into the bottom of narrow channels that are called gastric pits. And the secretions from several gastric glands are going to flow into each of these gastric pits and are then going to flow into the lumen of the stomach. So taking a closer look at these gastric pits and these gastric glands, they are going to be made up of several different types of cells that are found in the stomach. The gastric glands are going to contain three main types of exocrine glands, or exocrine gland cells that are going to secrete their products into the stomach's lumen. Right, these cells are the mucous neck cells, the chief cells, and the parietal cells. Both surface mucosal cells and the mucous neck cells are going to secrete mucus. The parietal cells are also going to then produce something called intrinsic factor, and this is really important for the absorption of vitamin B12. They're also going to produce hydrochloric acid. Those chief cells, also called zymogenic cells, are also then going to secrete pepsinogen and gastric lipase. These are going to be both very important for the breakdown of some of these nutrients. And the secretions of the mucus, parietal, and chief cells are all going to cumulatively form the gastric juice. In addition, the gastric glands are also going to include a type of enteroendocrine cell. These are called the G cells, and these are going to be located mainly in the pyloric antrum and are going to secrete the hormones gastrin into the bloodstream. So looking at the mechanical digestion that's going to occur in the stomach, because this is a very important part of the mixing that's going to go on. So several minutes after food is going to enter the stomach, there are going to be waves of peristalsis. They're going to pass over the stomach every 10 or 15 or so to 25 seconds. Most waves are going to begin at the body of the stomach and are going to intensify as they reach the antrum. The fundus is going to be primarily for storage when we're just extra full. So we have the body, and then they're moving towards the antrum. All right. Each peristaltic wave is going to move those gastric contents from the body of the stomach down into the antrum, and this is a process known as propulsion. The pyloric sphincter is normally going to remain almost, but not quite all the way closed. So it's going to be just barely open. And this is because most food particles, this is useful because most food particles in the stomach are initially going to be too large to fit through that very narrow pyloric sphincter. They are going to be forced back into the body of the stomach in a process that's called retropulsion. This cycle of propulsion and retropulsion is going to be repeated until those food particles have been broken down into something that's small enough to pass through that narrow little pyloric sphincter. The net result of these movements, of these propulsion and retropulsion, is going to be that those gastric contents are going to mix very thoroughly with gastric juices, and this is eventually going to become reduced down to some sort of gross soupy liquid that's going to be called chyme. Once the food particles in the chyme are small enough, they're then going to be able to pass through the pyloric sphincter, and this phenomena is known as gastric emptying. Gastric emptying is a very slow process, however, and only about 3 millimeters of chyme are going to move through the pyloric sphincter at each time. So it's going to take a little while. Then moving on and looking at the chemical digestion that's going to occur in the stomach. Looking again at the process of chemical digestion in the stomach. Parietal cells are going to secrete hydrogen ions as well as chloride ions, and they're going to secrete them separately into the stomach lumen. That is going to result in the eventual formation of hydrochloric acid. Proton pumps that are powered by a hydrogen potassium ATPase are going to then actively transport hydrogen ions into the lumen while bringing potassium ions into the cell. At the same time, chloride and potassium ions are going to diffuse out of the lumen through the chloride and potassium channels that are found on the apical membrane. The enzyme carbonic anhydrase, which is going to be especially plentiful in parietal cells, is going to catalyze the formation of carbonic acid from water and carbon dioxide. As carbonic acid dissociates, it's going to provide a ready source of hydrogen ions for that proton pump, but it's also going to generate bicarbonate ions as well. As bicarbonate builds up in the cytosol, it's going to exit those parietal cells in exchange for chloride via a chloride bicarb antiporter that's found in the basolateral membrane. This is going to be right next to the lamina propria. The bicarb is going to diffuse into the nearby blood capillaries, and this alkaline tide, as it's called, of bicarbonate ions entering the bloodstream after a meal may be largely enough, or maybe large enough, that this is going to cause an elevation of blood pH slightly, and it's going to make urine more alkaline. The hydrochloride secretion by the parietal cells can also be stimulated by several different sources. We've got acetylcholine. This is going to be being released by the parasympathetic system, or parasympathetic neurons. There's also gastrin, which can be secreted by G cells, and then finally there's histamine. This is different than the kind that makes you sneeze, but this is going to be a paracrine substance that's going to be released by mast cells in the nearby lamina propria. Acetylcholine and gastrin are going to both stimulate parietal cells to secrete more hydrochloride in the presence of histamine. So in other words, histamine is basically acting synergistically, and it's going to enhance the effects of acetylcholine and gastrin in the stomach. Receptors for all three substances are also going to be present on the plasma membrane of those parietal cells. You can see those here. So, moving forward continuing with our chemical digestion in the stomach, this strongly acid fluid that's going to be found in the stomach is going to kill any sort of microbes that are in food. This is a good thing. Generally helps to prevent food poisoning for the most part, not always. Hydrochloride is also going to partially denature the proteins that are in the food, and it's going to stimulate the secretion of hormones that are going to promote the flow of bile and pancreatic juices from other parts of the digestive system. This is going to help with digestion as well. The enzymatic digestion of proteins is going to begin in the stomach. The only proteolytic or protein digesting enzyme that's found in the stomach is called pepsin, and this is going to be secreted by the chief cells. Pepsin is going to sever certain peptide bonds between amino acids, and it's going to cause them to break down, breaking up that protein chain, going from many amino acids into a smaller peptide fragments. Pepsin is going to be most effective in the very acidic environment of the stomach, somewhere around pH of 2, and it's going to become inactive as the pH increases, so the environment becomes less acidic, more basic. So you might be wondering, what is it that's keeping this pepsin from actually digesting the proteins that are going to make up the cells that line your stomach, while it's still giving them the ability to digest food? Well, here, let's look at that. So pepsin is going to be secreted in an inactive form called pepsinogen, and in this form, it's not going to be able to digest any of the proteins that are found in the chief cells that produce it. Pepsinogen is not going to be converted into an active form, pepsin, until it comes into contact with the hydrochloric acid that's secreted by those parietal cells or other active pepsin molecules. So that additionally, the stomach epithelial cells are going to be protected from those gastric juices by a layer that's anywhere between 1 to 3 millimeters thick of alkaline mucus that's going to be secreted by surface mucus cells as well as mucus neck cells. And this is going to form a protective layer to keep that pepsin from basically eating your stomach from the inside out. So that's important. So another enzyme that's found in the stomach is called gastric lipase. This is going to split triglycerides, aka fats and oils, into the fat molecules into the fatty acids and monoglycerides that make up those fat molecules. This enzyme, which is going to have a relatively limited role in the adult stomach, is going to operate best at a pH of between 5 and 6. So more important than either lingual lipase or gastric lipase is going to be pancreatic lipase. And this is an enzyme that's secreted by the pancreas into the small intestines. The stomach wall is also going to be permeable to most substances. However, the stomach wall is impermeable to most substances. However, some water, electrolytes, and certain drugs, especially aspirin, as well as alcohol, can be absorbed through the stomach lining. This is why we have something called enteric-coated aspirin, because absorbing this aspirin, which is really quite acidic, can really irritate your stomach lining. So enteric-coated aspirin essentially has a coating on it that prevents it from being digested or absorbed in the stomach, and is then absorbed later on in GI tract somewhere in the small intestine. This whole absorption of certain things in the stomach is also another really important reason why you shouldn't drink on an empty stomach, and we'll talk a little bit more about that later on in the lecture. So, overall, looking at the functions of the stomach. 1, it mixes saliva, food, and gastric juices into a lovely sludgy slurry called chyme. It's going to serve as a reservoir for food before it's being released into the small intestine. It's going to secrete gastric juices, which contain hydrochloric acid, pepsin, intrinsic factors, and gastric lipase. And it's also going to secrete gastrin into the bloodstream. So here we have a handy table looking at the summary of the digestive activities that are going to occur in the stomach for later use in studying. Next up on the digestive tract, we have the pancreas. This is going to be a retroperitoneal gland that's going to lie posteriorly to the greater curvature of the stomach. You can there you see it right there. There's our pancreas. It's a goofy looking thing. The pancreas is going to consist of a head, a body, and a tail, right? The head, the body, and the tail of the pancreas. This is usually going to be connected to the duodenum of the small intestine by 2 ducts. The head is going to be the expanded portion of the organ, and it's going to lie near the curvature of the duodenum. And superior and to the left of the head is going to be the central body tapering to the tail. You can see that here. So the pancreas is a gland that's responsible for a variety of different things. It's going to lie posterior to the stomach, like we've just said, and it's going to produce enzymes that are responsible for digesting starch, aka pancreatic amylase. It's also going to produce enzymes that digest proteins called trypsin, chymotrypsin, and carboxyl peptidase. And then it's also going to secrete enzymes that digest fats called pancreatic lipase, as well as nucleic acids. These are going to be called ribonucleases and deoxyribonucleases. The pancreas is going to contain sodium bicarb, and this is going to convert the acid that's in the stomach or the stomach contents to a slightly alkaline pH, somewhere between 7 and 8. And this is going to effectively halt the stomach pepsin's activity, and it's going to also promote the activity of pancreatic enzymes. The pancreas is going to empty its contents into the duodenum, which is the very beginning part of the small intestine. So, looking at the anatomy of the pancreas. Pancreatic juices are going to be secreted by exocrine cells into small ducts that are ultimately going to unite to form 2 larger ducts. These are going to be the pancreatic duct and the accessory duct, and these are going to convey the secretions into the small intestine. The pancreatic duct, also called the duct of Wirsung, is the largest of the 2 ducts, and the pancreatic duct is going to join the common bile duct from the liver and the gallbladder, and it's going to enter the duodenum as a dilated common duct. This is called the hepatopancreatic ampulla, also called the ampulla of Vater. The ampulla of Vater, or this ampulla, is going to open on an elevation of the duodenal mucosa. This is called the major duodenal papillae, or papilla, and this is going to refer to, and this is going to lie inferior to the pyloric sphincter that's found in the stomach. The passage of pancreatic juices as well as bile through the hepatopancreatic ampulla into the duodenum of that small intestine is going to be regulated by a mass of smooth muscle that is going to surround the ampulla, and this is known as the sphincter of the hepatopancreatic ampulla, also called the sphincter of Oddi. It's a much nicer, easier name there than sphincter of the hepatopancreatic ampulla, but both names are acceptable. The major duct of the pancreas, the accessory duct, also called the duct of Santorini, which I think is kind of a fun name, is going to lead from the pancreas and it's going to empty into the duodenum superiorly to our hepatopancreatic ampulla. Looking at the histology now of the pancreas. This is made up of small clusters of glandular epithelial cells. About 99% of these clusters are called acini. These are going to constitute the exocrine portion of the organ. The cells that are going to lie within those acini are going to be a mixture of fluid are going to secrete a mixture of fluid as well as digestive enzymes called pancreatic juice. Those remaining 1% of the clusters of cells are called pancreatic islets. These are going to form the endocrine portion of the pancreas, and we talked about those previously in our endocrine chapter. These cells are going to secrete the hormone glucagon, as well as insulin, somatostatin, and pancreatic polypeptide. Those are being secreted by our pancreatic islets here, which we can see. But the vast majority of the pancreas is devoted to these acini, which are going to be the exocrine portion of the pancreas. So, moving on then to the liver and the gallbladder, just a kind of silly little cartoon for your enjoyment there. The liver is going to be divisible into a left and a right lobe, and it's going to be separated by the falciform ligament, which we mentioned earlier in this lecture. The liver is going to be the heaviest gland and the second largest organ in the body, the largest organ in the body being the skin. So this is the second largest. So it's a pretty sizable chunk of tissue here. The lobes of the liver are going to be made up of lobules, and these are going to contain hepatic cells, also called liver cells or hepatocytes. They are going to also contain bile canaliculi and hepatic sinusoids. So let's start off by looking at those hepatocytes. These are specialized epithelial cells, and their main function is they're going to be the main functional cells that are in the liver. So they're going to perform a wide array of metabolic, secretory, and endocrine functions. Hepatocytes are going to form these complex three-dimensional arrangements that are called hepatic lamella. Hepatic laminae. These hepatic laminae are going to be pleats of hepatocytes. They're going to be one cell thick and are going to be bordered on either side by the endothelial-lined vascular spaces, and these are called hepatic sinusoids. Grooves that are in the cell membrane between these neighboring hepatocytes are going to provide space for the canaliculi into which the hepatocytes are going to secrete bile. Bile is a yellowish, kind of brownish or sort of olivey greenish liquid that's going to be secreted by those hepatocytes, and it's going to serve as both an excretory product as well as a digestive secretion. So it's kind of multifunctional there. Next we have the bile canaliculi. These are small ducts that are going to reside between those hepatocytes, and these are going to collect the bile that's produced by those hepatocytes. From those bile canaliculi, this bile is going to pass into the bile ductules, and then it's going to end up in the bile duct. These bile ducts are going to merge and are eventually going to form a larger right and left hepatic duct, and these are going to unite and exit the liver as the common hepatic duct. The common hepatic duct is going to join the cystic duct that's going to come from the gallbladder, and this is going to form the common bile duct. And then from there, bile is going to enter the duodenum of the small intestine, and it's going to play a role in digestion. Within those hepatic sinusoids, these are going to be highly permeable blood capillaries that are going to be between the rows of the hepatocytes, and these are going to receive oxygenated blood from the branches of the hepatic artery, as well as nutrient-rich deoxygenated blood from branches of the hepatic portal vein. Hepatic sinusoids are going to converge and are then going to deliver blood into the central vein. From the central veins, the blood is going to flow into the hepatic vein, which are going to drain into the inferior vena cava. In contrast to blood, which is going to flow towards the central vein, bile is going to flow in the opposite direction. Additionally, thing present in the hepatic sinusoids are going to be fixed phagocytes. These are called stellate reticuloendothelial cells, also called Kupffer cells. And these are going to be hepatic macrophages that are going to be there to destroy any sort of worn out or broken down white or red blood cell. They're also going to remove foreign matter that will show up in the venous blood that's drained from the GI tract. So last but not least for this lecture today, we have the gallbladder. So the gallbladder is going to be made up of a couple of different parts. We've got the fundus, which is down here, the body, and the neck of the gallbladder. The fundus is going to project inferiorly, while the body and the neck are going to project superiorly. So it's kind of a little curved shaped thing. You can see by the way it's shaped there. The mucosa of the gallbladder is going to be made up of simple columnar epithelium that are going to also be arranged in rugae. There is actually no submucosal layer in the gallbladder, so it's going to be a little bit different in that way. And then the smooth muscle of the muscularis is going to be responsible for ejecting bile into the cystic duct, which is going to be what's going to connect, which is the duct that's going to connect to the gallbladder and empty those ducts, or empty that bile into the intestines. So the outer layer of the gallbladder is going to be the visceral peritoneum, and the gallbladder is responsible for storing and concentrating bile until it can be used, or until it's needed in the small intestines. So looking at the role of the pancreas, the liver, and the gallbladder all coming together and how they work all together as part of this digestive tract real quick. The liver is going to make bile, which is going to be important for the emulsification or the breakdown of fats. The gallbladder is going to store that bile until it's needed, and then the pancreas is going to secrete pancreatic juices that are really important for digestion and buffering. So looking at the flow of the various ducts that are going to carry bile from the liver and the gallbladder, and as well as the pancreatic juices, all of these are being secreted, are producing products that are going to be secreted into the duodenum of the small intestine. So we've got our liver is secreting bile, and this is going to drain into the right and left hepatic duct, which are going to flow into the common hepatic duct from the liver. Right? This is then going to enter into the common bile duct, which will then be emptied into the duodenum. Right? Simultaneously then, our cystic duct from the gallbladder is going to be emptying its contents into this common bile duct, but it's also going to receive bile from the common bile duct as well that can be concentrated and stored until it's needed at a later time. And then the pancreatic duct is going to be emptying its pancreatic secretions directly into the duodenum there. Looking then at the blood supply of the liver, this is going to receive blood from two main sources. First is from the hepatic artery, and this is going to contain all of the oxygenated blood. Right? You can see that here. Right? And then from the hepatic portal vein, the liver is going to receive deoxygenated blood. It's going to contain newly absorbed nutrients, as well as any kind of drugs or possible microbes or toxins from the gastrointestinal tract. So we could see that here. The branches of both the hepatic artery and the hepatic portal vein are going to carry blood into the hepatic sinusoid, and this is where oxygen, most of the nutrients, as well as certain toxins are going to be taken up by those hepatocytes. From there, the products that are manufactured by those hepatocytes, as well as any nutrients that are needed by other cells in the body, are going to be secreted back into the blood, which is going to drain into the central vein and is eventually going to pass into the hepatic vein. All right? Then from there it will move back to the inferior vena cava into the right atrium of the heart, where it will continue its journey through the circulatory system. So most of the blood from the gastrointestinal tract is going to pass through the liver, and it's going to do so as part of the hepatic portal circulation. So the liver is often going to be the site of metastasis for various types of cancers that originate in the GI tract. So that's an important thing to just kind of keep in mind. So finally, looking at the functions of the liver and gallbladder together, these two things are going to be important for carbohydrate, lipid, and protein metabolism. Also going to be involved in processing drugs and hormones, as well as bilirubin secretion, bile salt synthesis, storage of glycogens, vitamins, and minerals, as well as phagocytosis by those Kupffer cells, as well as vitamin D activation. So all of these things are going to be very important functions that those liver and gallbladder are going to achieve in one way or another. So up next, last but not least, we have to talk about jaundice. So jaundice is going to be a kind of yellowish discoloration of the sclera, with like those white parts of your eyes. It also could show up as a yellowish discoloration of the skin and the mucous membranes. And this is going to be due to a buildup of a yellow compound called bilirubin. So bilirubin is going to be formed from the breakdown of the heme pigment, the oxygen binding pigment in red blood cells. So it's gonna, when we have a breakdown of these heme pigments in old red blood cells, this is going to be transported to the liver, where it's processed, and it's eventually going to be excreted into the bile. So there are three main categories of jaundice. We have our prehepatic jaundice, and this is going to be due to an excess production of bilirubin. We have hepatic jaundice due to congenital liver failure, cirrhosis of the liver, or hepatitis. And then we have extra hepatic jaundice, and this is going to be due to a blockage of the bile drainage by either a gallstone or possibly by cancer of the bowel or the pancreas. So those are the kind of three types of jaundice that we see in adults or in fully grown people. However, in newborns there is something called neonatal jaundice. This is going to form because the liver of a newborn is going to function rather poorly at first, and so many babies are going to experience some sort of mild form of jaundice that we call neonatal jaundice, also called physiological jaundice. And this is going to disappear as the liver matures and is able to remove that bilirubin from the body or from the system. In the meantime, though, while you're waiting for your baby's liver to fully kick into gear, jaundice is typically treated by exposing infants to blue light. This is my daughter Maggie here, and this is when she was a couple of days old, and she had to sleep on this crazy sun tanning machine that exposed her to blue light, which would help to convert that bilirubin into a substance that the kidneys could excrete. And this is because bilirubin is photosensitive, and exposure to blue light causes it to break down. Back in the day, before they had these crazy sunbed systems here, they used to set babies outside or in the window with just a very few clothes on, and the exposure to the sunlight would also cause the breakdown of the bilirubin, helping that jaundice to clear up while you were waiting for baby's liver to kick into gear. So just kind of an interesting side note on the role of the liver in the body. So that is all we have for this lecture. I've included the Wiley Plus animations links here. These are available in the regular PowerPoint slide. As always, if you have any questions, comments, or concerns, please feel free to get in touch with me in email or during the next class. So until then, I'll see you next time.